Hi, my name is Rosalind Franklin, and I am known for my work with X-ray diffraction analysis to determine the structure of DNA. So I studied physical chemistry at Cambridge University, and in 1945, I earned my PhD after working with the British Coal Utilization Research Association, where I studied the properties of coal. After this, one of my mentors from college, Adrian Wheel, helped me get a position with Jacques Mering at his lab in Paris, and that's where I learned the technique of X-ray crystallography, which is sometimes called X-ray diffraction analysis. After working with him in Paris, I was a really good candidate for a fellowship at King's College, known as the Turner and Newell Fellowship, and I worked in the biophysics unit at King's College in London, and that's where I did all of my research on the structure of DNA. Initially, John Randall, who was in charge of the biophysics unit at King's College, wanted me to work on analysis of proteins using my X-ray diffraction analysis technique, but at the request of the assistant lab chief, Maurice Wilkins, I was asked to work on DNA instead. I didn't know it at the time, but Maurice Wilkins was actually kind of wanting to work with me on my research. But because I didn't know this, I chose to work with Raymond Gosling, who was a graduate student, and my relationship with Maurice Wilkins was kind of rough after this miscommunication. X-ray crystallography is a technique used to determine the structure of a substance, usually a protein or a macromolecule. X-rays are ideal to use for these experiments because the wavelength of an X-ray is really similar to the distance between atoms of a crystal. So when an X-ray encounters an atom, energy is absorbed by the electron, but it's not enough energy for the electron to release. So the energy is released, and it's released at a different angle than from where it came. So this is known as elastic scattering. In a crystal, the uniformly spaced atoms form planes that are separated by well-defined distances, and when the atomic planes are exposed to an X-ray, the light is scattered at a very specific angle. And those angles can tell us a lot about the molecular shape of the sample. For example, this is known as photo 51. This is from my research on DNA, and we can learn a lot from this image. I guess the most obvious is this very clear X shape that you see is a telltale sign of a helical structure. And the angle here gives us the angle, or it indicates the steepness of the angle of the helix. This image also showed that the phosphorus groups were on the outside of the helical structures rather than on the inside, which was previously hypothesized. Because before doing this experiment, we already knew that phosphorus causes intense reflections under an X-ray. And finally, I was also able to identify 10 base pairs for every turn of the helix from this image. I worked with Raymond Gosling, who was a graduate student during this research project, but I was under the supervision of John Randall and Maurice Wilkins, who I previously mentioned. You may also be familiar with James Watson and Francis Crick, who were also working on a theoretical model of DNA at the time at the Cavendish Laboratory at Cambridge University. In 1953, January of 1953, Wilkins showed Watson and Crick my images, including photo 51, along with some of my unpublished research, and Watson and Crick went on to publish their findings in a journal called Nature in April of that year. Wilkins, Watson, and Crick went on to win the Nobel Prize in 1962 for the discovery of the structure of DNA without acknowledging any of my contributions. Much of my education, including my college years, were set against the backdrop of World War II. So I lived in England for most of my life, and that was kind of, World War II was happening all around us. A lot of my professors were impacted by this, so some of them had to flee the country. Some of them who were German were detained. And then I also had some new professors come into town because they were refugees. For example, my mentor, Adrian Wheel, who I mentioned a little while ago, he was a refugee from France. So I'm kind of grateful for that aspect of it. Otherwise, I mean, just thinking about London in general, like you walk around town and, there's still destruction from the war, like damaged buildings that haven't been repaired yet because we don't have the funds to repair them yet. Taxes were also really high during this time. Rationing still happened, like way after the war ended. And also there was a boom in industrialization, which was good, but it also meant more pollution. And in 1952, there was the Great Smog, which lasted for 5 days and resulted in the death of about 4,000 people. So yeah, a lot was going on around this time. Discovering the structure of DNA opened a lot of doors in the scientific community to study genetics. Generally speaking, when you understand the structure of something, it makes it a lot easier to understand how it functions. And so this was really helpful. In the physics community, I was using X-ray crystallography at a time when not a lot of people were using it. It hadn't been around for a terribly long time. And I was using it on really complex structures. So my work contributed to the foundation of the technique, and I found a lot of ways to make it a lot more precise. For example, I would introduce hydrogen into the sample because hydrogen doesn't have a lot of electrons, and so there was a lot less interference in the results when I introduced more hydrogen. After Watson and Crick published their findings in 1953, I transferred my fellowship to Birkbeck College, where I studied viruses in plants, particularly the tobacco mosaic virus. And one of my biggest findings was that the genetic material RNA is actually embedded in the inner wall of the protein shell of the plant cell. And unfortunately, around this time, I also was diagnosed with ovarian cancer. And I only lived for about 2 years after that diagnosis. In the last couple of years of my life, I was asked to speak a lot at conferences, and a lot of people think that if I hadn't died so young, I probably would have gained a lot more professional recognition for my work in the scientific community. Oh, so fun fact. When I was a girl, I attended St. Paul's School for Girls, and while I was there, my music teacher was Gustav Holst, who you may know because he was the composer of an orchestral suite called The Planets, which, if you haven't heard it, it's a really cool piece. Unfortunately, I was not great at music, and I think the best compliment he ever gave me was that my singing was almost in tune.